Hello, my name is Rahul Mehendale, and I lead new business creation within Deloitte's life sciences and healthcare practice. With me is my colleague Juergen Klenk, who is our precision medicine leader. We are here today to examine the future of biomedical research. We are going to look both at how biomedical research has been conducted to date and how the landscape is changing, as well as a potential solution that we are using to address this innovative field. Biomedical research is at a unique juncture. It is increasingly turning to data science to make new discoveries for treatments. Let me give you three examples of drivers that are causing this change and the resulting needs for data science. First, there are large volumes of data now available, orders of magnitude more than before, that need to be stored for efficient use. This includes the traditional sets of science-generated data, such as genomics data, imaging data, clinical data, but also patient-generated data from wearables and environmental data and exposure data. These data are now being collected in ever-increasing quantities, and there is an expectation that if they can be easily stored and analyzed, they can help drive new discoveries and treatment options. Thus, there is a huge need for storage and efficient search capabilities for biomedical data. Second, there is an interest in driving deeper into precision medicine by using artificial intelligence. Researchers want to understand how the body functions at all levels, starting at the molecular level, then at the cellular level, and the organ level. Take cystic fibrosis as an example. Researchers found a correlation between the CFTR gene and the disease, leading to the understanding that a mutation in the CFTR gene was the cause of cystic fibrosis. From this discovery, a new treatment could be developed, and 2 years ago, the FDA granted a breakthrough therapy designation to a particular gene therapy that addressed the mutated gene. However, in most cases, the relation between genetic mutation and disease is much more complex. It is not a one mutated gene causes one disease situation. So researchers need to go further. They need to understand how the body functions at the molecular level, looking at all genes and proteins in action within cells and organs. This will require the development of new artificial intelligence methods that can analyze all of the data available. Thus, biomedical research is becoming a huge computational task. Third, there are an estimated 7,000 rare diseases, of which only 500 have treatments and even fewer have cures. Thus, there's an urgent call to researchers to share all of their data to rapidly advance the discovery of new treatments. While each of these 7,000 diseases may be rare individually, the total tally affects a significant number across the population. To note, we learned that cancer is not just one disease, but rather it is a collection of many different types of cancer, some of which are very rare. We need to understand what is happening to the body with each of these different cancer types in order to find cures. By sharing their data with the research community, there is an opportunity to perform new analysis and make new discoveries. Let me summarize. To make new discoveries and find new treatments, biomedical researchers will need to be able to store, analyze, and share all of their data at scale. We are now going to look at a particular use case that focuses on the necessary steps to share data without compromising privacy. The biomedical data we use for research are collected from humans, and ensuring the privacy of these data donors is critical. Without it, we would put at risk the trust given to scientists by these data donors. The HIPAA privacy rule establishes the conditions under which protected health information, or PHI, may be used or disclosed for research purposes. In short, HIPAA requires that all PHI be de-identified before data are shared. In addition, other laws require that all personally identifiable information, or PII, also be de-identified. Our use case focuses on the de-identification of imaging data for cancer, cardiovascular, and pulmonary research. A critical step before these images can be shared with the research community. On this slide, you will see a pair of lung X-rays. Today, most CT scans, MRIs, and X-rays use the DICOM standard, which stands for Digital Imaging and Communications in Medicine. DICOM data has a header which includes PHI and PII data. See the red boxes on the top left and right. In addition, the PHI and PII can be found on the image itself. See the red box at the bottom. We are working with our client on a pilot to ensure their patient's data is de-identified by removing, masking, or encrypting the sensitive PHI-PII data before images are shared. To do this, we are using Google's Healthcare API as an out-of-the-box solution. In our pilot, we are working to answer two questions. How reliable is the de-identification process? And what is achievable throughput of the process? This is important as we are working to do this at scale. As you can see, de-identifying data just from images is a large undertaking, and this is only one of many steps that must be taken to prepare data for large-scale analytic efforts. If we take a step back and look at all the steps required for biomedical research, we realize that what we need is an efficient solution that provides the biomedical community with the ability to store, analyze, and share their data at scale. We will now discuss how we can build such an environment that meets these needs on the Google Cloud platform. However, we should note that a technical solution alone will not be sufficient to create a thriving ecosystem of biomedical research that widely shares their data. There are several non-technical challenges that must be overcome, but they are beyond the scope of this presentation. I'm now going to hand over to Juergen to walk us through the details of the GCP-based solution.
Thanks, Rahul. To start, let's examine the principles of a biomedical research data science solution. As Rahul mentioned, there are three key functional requirements: the ability to effectively store and manage data, the ability to analyze data at scale, and the ability to share data with the community and effectively use the shared data. Let's look at each of these three in turn, starting with data organization. A foundational principle for organizing biomedical data is the concept of FAIR data. FAIR stands for findable, accessible, interoperable, and reusable. Let's look at what each of these mean. F for findable, A for accessible, I for interoperable, and R for reusable. Starting with findable. This means that all data must be annotated with metadata. That makes the data searchable. This must be possible for all data types. The data should be stored in a data lake, and we need a proper metadata repository. Data that is newly ingested should ideally be annotated with metadata in an automated or semi-automated way to allow data ingest at scale. Finally, we need an efficient search mechanism that lets us discover data of interest. This must be possible for all data. Today, most data sit in silos that cannot be searched from the outside. The next one is A for accessible. This means that there must be an easy way to request access to data that has been discovered and is of interest. Ideally, we don't want to move the data around, so data access really means that I can use the data to compute on it and quite possibly compute across many data sets that may reside in different locations. What we have today, again, is still mostly data silos, where it is impossible to run algorithms across the data sets. The third FAIR data concept is I for interoperable. Now, this is where data standards come into play. Unfortunately, in biomedical research, there's a myriad of standards. At a minimum, data should adhere to a common standard, and we need mappings between the most common standards. And yes, by data standards, I also mean semantic standards such as ontologies and vocabularies, not just syntactic standards. I is probably the hardest of the four FAIR principles. Finally, R for reusable. This has to do with data use agreements and consents that accompany the data. It must be possible to trace what data are used for, so that any misuse can be discovered and appropriate measures to protect the data and their human data donors can be taken. When it comes to the second requirement, powerful data analysis, we have to realize that algorithms are actually just data as well, and thus their code should also follow FAIR principles, because in the end, we not only want to share the raw data, but also our analysis methods in order to accelerate innovation. We need an easy-to-use data science environment that allows us to scalably compute across all of the data of interest. Furthermore, we want to be able to reuse already existing algorithms. Many of these are encoded in the form of services that are natively available on the Google Cloud platform. The previously discussed de-identification service is such an algorithm, and we need to be able to access it programmatically through its API in an easy way. Many other algorithms are bundled into libraries. This could be machine learning and artificial intelligence methods for image analysis or for a genomic study. Again, these algorithms or libraries must be easily accessible from within the data science environment. The last requirement is data sharing. We already discussed this as part of the FAIR principles, but to reiterate, sharing must be possible not only within teams, but also across teams and even between institutions. If we follow these guiding principles when building a solution on the Google Cloud platform, we will have enabled the biomedical research community to focus on what they are best at, doing their research without having to worry about the underlying technology, and hopefully be able to accelerate discoveries and new treatment options for the many untreatable diseases that ail us. We have developed a GCP-Native reference implementation for a biomedical research solution that follows the principles we just discussed. You can see some screenshots on the right, and I will show an architectural diagram later on. The data types we have to handle include clinical data in the form of electronic health records, often now following the FHIR standard, genomic data, medical and scientific imaging data, radiology images follow the DICOM standard, but pathology images do not consistently follow that standard. And we have also handled medical supply chain and public health data labs, mostly as part of our work for the COVID-19 Healthcare Coalition, which is a pro bono effort of almost 1,000 organizations coming together to share data and perform studies to combat COVID-19. We're also supporting a variety of analytic methods. Researchers often prefer to use a notebook-like environment to code their methods and workflows, specifically Jupyter notebooks that can be coded in R or in Python, to select datasets and build cohorts, but also to generate downstream analyses. BigQuery can be easily used. We also discussed access to microservices via their APIs. The Google Cloud platform, through its many healthcare and biomedical products, offers access to many services ranging from genomic analyses to data mapping to image analytics to advanced machine learning and artificial intelligence and more. These are powerful tools that we can bring to the researchers' fingertips. And finally, we offer the ability to reuse existing workflows through sharing, enabling true collaboration in method and algorithm development. Now let's take a closer look at the solution. You can see here two screenshots. The left screenshot shows the home screen where you can see all of your projects listed. Projects are the big tiles on the screen which show the project name, a short description, and the approved users. On the right screenshot, we opened one project. At the top, you can see the project information again. On the left is the metadata repository, which is searchable and where you can select data for use as well as share data with others. And on the right side are the workspaces where you can launch Jupyter notebooks to perform your analyses. What's important is that this user interface is intuitive and takes away from the complexity of the Google Cloud platform itself. If I showed the GCP home screen to a biomedical researcher, they would be overwhelmed by the choices of tools and often not understand how to use them. Our user interface provides them with an intuitive screen that they can understand how to navigate and perform their work, all the while using the full power of GCP behind it. Let's take a look at the architecture. I'll start on the left side. This is where we receive data from systems such as microscopes or other devices that can stream their data right into our environment. We can use a command line interface to push the data with a defined frequency, or we can manually load the data. The ingest process uses Cloud Network and Cloud Data Fusion to push the data into a Google Cloud Storage bucket. The metadata repository near the middle of the slide serves as an environment to store all metadata associated with the collected data. Upon data ingest, the data can be inspected for tags, for example, fire or DICOM tags, that can be used to automatically generate metadata. We use services provided by the Google Healthcare API to the left of the metadata repository to perform initial transformations of the data, such as de-identification. This requires temporary placement of the data into a fire or DICOM store where the de-identification service is being performed. We have a data sharing capability that invokes PubSub, Firebase, and can leverage services such as Google's Data Marketplace to make data discoverable. For search, near the top of the slide, we use Apache Solr. And to the right, we have notebook services that utilize GCP's Jupyter Lab to launch notebook services. In these notebooks, all the way on the right, in the second box from the top, the researchers can code and execute their algorithms. They can also access GCP services through what we call convenience methods, which are wrappers around the APIs so that researchers don't have to learn the detailed command syntax of an API call, but instead can use a very simple and intuitive command such as de-identify with some simple arguments. For security, we use the native GCP security layer, which provides identity and access management and offers a very granular role-based data access. Cost management is also built in to allow researchers to see the cost of a service before they use it. Above all, we strive to follow the open community's best practices. Data will not end up in a proprietary silo. Rather, it sits in a GCS bucket and can be accessed and reused from any other environment. Similarly, compute uses native GCP components, which will work across datasets stored in GCS. What we hope to illustrate with this reference implementation is to demonstrate how we can stop building more proprietary data silos in biomedical research that will just prevent the community from collaborating efficiently. If we follow the standards and best practices we laid out, solutions like our reference implementation will enable the construction of a collaborative biomedical research environment that can help researchers accelerate discoveries and treatment. Now, I'll hand it back to Rahul to close out our discussion.
Thanks, Juergen, and thanks, Google, for the opportunity for this discussion today. In summary, we have reviewed how biomedical research is transforming into a data science. We discussed the principles of how biomedical data can be effectively managed and analyzed in the Google Cloud platform, and we showed you a reference implementation. In addition, we discussed how Google Cloud Platform allows us to do biomedical research in a highly scalable and cost-effective way in the cloud. And finally, and most importantly, a cloud-native solution for researchers to collaborate and share their data with one another is vitally important to accelerate scientific discovery and find new treatments for the many still untreatable diseases. If you have any questions on this presentation or the content, please feel free to reach out to us directly. We have included our contact information on this slide. Thanks again for your time and interest.